Hi, welcome to my last lecture of this semester. We're going to talk about development of the eye and the ear, and also we're going to talk about some mechanoreceptors, proprioceptors, and other sensory cells, the primary and secondary sensory cells in the end of my lecture. So let's start with the development of the eye. Eye is a very complicated structure, as you know, is made of like three basic layers, the tunica fibrosa, the vasculosa, and nervosa. You should already know that tunica fibrosa is composed of sclera on the surface, the white structure of the eye, the sclera, and cornea, which is transparent. Yes, the cornea, the tunica vasculosa is actually made of three parts: the iris, the corpus ciliary, or the ciliary body, and just put number 2 here, and the third number goes to choroidea. So three structures make up the tunica vasculosa, and then we have tunica nervosa, which is composed of retina. Yes, and it's like that. And then we have lens crystallina, which is actually from the outer ectoderm. So lens is developed from the ectoderm, and we're going to talk about it now. So what is going to happen? It's going to, like the prosencephalon. The prosencephalon is going to create an outpouching, which is called the optic cup, but first we will create an optic vesicle. Yeah, first we have an optic vesicle, this outpouching. There is a sulcus on the tip. This outpouching is an optic vesicle, and there's still like communication with the inside of the prosencephalon. So here in the forebrain, there is an invagination of the, or I should say evagination, but it really looks like this, yeah, that we have the forebrain, which is lined with neuroectoderm. Neuroectoderm is lining this forebrain in the inside, and we have these optic grooves. So we created optic outpouchings like this from the forebrain, which are lined with neuroectoderm. We still don't have the closure of the cranial neuropore, but in 24th to 25th day, there is a closure of cranial neuropore, so it will close eventually. And in the forebrain, we have something called eye field, which is very important for the signalization pathways, not so important for the knowledge of histology, but you should know that we have an eye field on the top of the prosencephalon. The prosencephalon, the forebrain, and that this eye field is important in signalization pathways. That we have a PAX6 here. This one is important for the eyes. So once again, we have a forebrain here, the cavity is inside, and these outpouchings are optic vesicles here. They are getting closer and closer to the surface ectoderm, as you can see, these outpouchings. And when they are very close enough, the ectoderm on the surface will react somehow. Yes, because something is touching the ectoderm. There is a signalization going on. So ectoderm will actually react to this optic vesicle from the cavity of the forebrain and will get thicker. So cells in here on the surface ectoderm will proliferate. And what are they starting to do? They will start to invaginate. Yes, they will start to go closer and closer to the optic vesicle here. It's like optic vesicle wanted to be very close to the ectoderm, and ectoderm will react. Okay, you want to be close to me, so I want to be close to you. So come on, yes, it will invaginate, and it will really get so close to the optic vesicle that this optic vesicle will react and it will invaginate the inner layer in here, and we will create something called optic cup. Yes, because it really looks like a cup. We have inner surface and outer surfaces of optic cup. And this is very important because some structures will develop from the ectoderm, which is proliferating and invaginating because of the optic cups, and some structures will eventually develop from the optic cup. And don't forget, there is also this mesenchymal tissue all around, and this mesenchymal tissue comes from ectomesenchyme. Is really of neural crest origin, together with mesoderm of the head. Yes, so normal mesoderm and neural crest cells, which form the mesenchyme, and also mesoderm form mesenchyme, will create some structures in the eye. Yes, but remember this thing, this is very important, that we have optic cups and lens placket, which is lens placket is really the term for the thickening of the surface ectoderm. And this outpouching of the forebrain is very important in development of lens placket, because when there is no outpouching, the lens placket is not going to develop. Yes, we need this touching, we need this signalization going on here, yes? So something needs to happen in this place. So lens placket will eventually form the lens. Yes, that's like kind of obvious, but the optic cup is kind of different and more difficult. You can see that on our pictures. No, these are not our pictures, but we cannot see that on our slide, but I wanted to show you like how does it look like in really on the histological specimens, that we have the cavity of the forebrain here. The forebrain is lined with neuroectoderm, and you know something about neuroectoderm, at least I hope so, because all neurons and glial cells are developed from the neuroectoderm, except for microglial cells, which are from mesenchyme, but all glial cells and neurons, except for microglial cells, are developed from this neuroectodermal epithelium, which is a pseudostratified epithelium. So outpouching of this cavity will form the optic cup, which get very close to surface ectoderm, and the surface ectoderm will form the lens. Yes, I can't stress that more enough. It's enough for me to say it like 3 times in a row, so I hope you will remember. And let's go somewhere else. And this is once again the same thing here, but the optic cup is still connected to the cavity. Yes, you can see the connection. It's still connected to the cavity, and this connection is called optic stalk. Yes, it's continuous with the wall of the forebrain. It's an optic stalk, and we have some cavity inside the optic cup. This cavity is inside the optic cup is called intraretinal space. Intraretinal space. Why do we call it intraretinal space? Is that it's inside the retina. Inside the retina. And as you probably know the structure of the eye, like where is the space inside in the retina? Like there is no space in there. So these two layers, the outer layer of the optic cup and inner layer, will get close together to close, yes, this intraretinal space. So this space in between these two layers will eventually close, and we will form the future normal retina. So please be aware that we differentiate inner layer of optic cup, which is a primordium of neural layer of retina, and we differentiate outer layer of optic cup, which will form the retinal pigmented epithelium. As you know, retina has like 10 layers, like many layers. There are many layers of retina. And the first layer, when we go from the periphery, the first layer is pigment epithelium. That's the first layer. Actually, this first layer, the pigment epithelium, will develop from the outer layer of optic cup. Isn't that crazy? And all other layers are from the inner layer of optic cup. Yes? So, and you should know that because the retina, the pigment epithelium, and the second layer of retina, which is made of rods and cones, are really actually they are from the same origin. Yeah, they are developed from the optic cup, but they are from different layers. Yes, and that is important. They are not connected to each other very firmly. So this intraretinal space doesn't actually exist in the normal retina, but in some trauma, when there is some disruption of the retina of the eye, these two layers can separate from each other, and there can be a space. Yes, and there is some kind of damage in the retina and disruption of the vision. I don't know if I explained it in the way that you can understand, but just remember the pigment epithelium isn't attached very firmly to rods and cones, and that might be a problem in future traumatization of the eye. Yes, there are no tight junctions in between, none of them. And we can see also in this picture the lens placode, which is invaginating and forming the lens. Yes, lens will form from the surface ectoderm. And we have also mesenchyme all around. These cells are mesenchymal cells. And you can say that, okay, they come from the neural crest because most of the tissues that are in your head come from neural crest, but there is a combination. It's like normal mesoderm and also mesenchymal cells, the neural crest cells. Yes, but mesenchymes mostly comes from neural crest. I will write it down for you to remember. Neural crest. Yay. Okay, so from now on, please remember there are like three origins of the eye. There is this surface ectoderm, there is neural ectoderm, and there is also neural crest. And also we need some factors, and you can see right here that there are many factors involved in eye development. The most important ones are sonic hedgehog, I would say, and this PAX6, which is important also, but you don't have to know about it very much. Just forget about it. Don't worry about it. So, we need some vascularization of the eye also. Yes, because many structures are developing here, they are getting bigger and bigger. So diffusion isn't really enough for this development. Cells are proliferating, so we need some supply during development only, because normally we don't have this very big artery that is going through the whole eye. We just have smaller arterioles and capillaries inside the wall of the eye, but we don't have an artery that is going just right straight through the eye. But during development, in this optic cup, there is a fissure created. Yes, so this is the fissure, and hyaloid artery is going from the ophthalmic artery. So we have the ophthalmic artery. The branch of ophthalmic artery is hyaloid artery, which is going through the fissure. It's going inside to the eye, and it's supplying the lens, the developing lens, which is here, the hyaloid artery. Very important artery, because sometimes in adulthood you can find remnants of the hyaloid artery. Yes, in the eyes. Because a hyaloid artery will eventually disappear. When the eye is fully developed, you don't need to supply lens anymore, because lens is fully developed, and the hyaloid artery will disappear in that case. So you can see the choroid fissure in here, right? It's called the choroid fissure. This is how it looks like in a cross section. And the hyaloid artery is in the middle. Once again, don't forget about inner layer and the outer layer. It's like here. The inner layer is much more thicker, but it makes sense because it's going to make like 9 layers of the retina, and the outer layer is thinner because it's going to give rise to pigmented epithelium. And we have intraretinal space in between, which is continuous with the cavity of the forebrain. And lens vesicle, because in some time, yes, we have, in this case, we have still connection with the surface ectoderm, but at some time when we want to develop like full lens, we want to separate the lens from the ectoderm, because your lens is not connected with the ectoderm, yes, so we need to separate it. So in this case, it's fully separated. We have the lens vesicle. We differentiate the anterior epithelium and the posterior epithelium, and you can see right now something is happening here. Yes, it's very surprising that something is happening in the posterior epithelium because these cells are much more longer, they are more cylindrical, and these are more cuboidal. So we're going to fill this lens eventually. And we have this lens vesicle, which is supplied with the hyaloid artery by the choroid fissure. So the hyaloid artery isn't actually going in from the inside of the brain, and somebody will think of it like that, but it's actually going from the outside, from the ophthalmic artery as a branch. Yes, and this is really how it looks like. So the fissure needs to close eventually, yes, because we need to form a pupil inside, yes, a pupil. I don't know how to pronounce that. Pupilla of the eye, yes, the hole like the side of the eye, yes. And the iris is going to form around. The cornea will be formed ventrally to the pupilla. Right behind pupilla we will form the lens, so there is this opening, the pupilla, but we need to close the fissure in order to create the round papilla, like this. So this fissure will close, and we will have the hyaloid artery inside. Hyaloid. Okay, let's go next. These are some pictures, very nice pictures. Here we have still a connection with the surface ectoderm, the opening, yeah, so you can see it's just invaginations. Cells are proliferating inside, and then we have the the lens vesicle, which is still empty on the inside, but that will change eventually. And we have optic cup. Optic cup with the outer layer and inner layer. Also the same picture in here, but it's really nice. I like it. So, but here you can really see, yes, the space, the intraretinal space. There you go. We cannot really see the connection from this side, but yes, just remember, intraretinal space is connected with the space inside the forebrain. This is very important. Okay, so what is going to happen? We are eventually forming the layers, all the layers. We can see some differentiation of the layers here in the retina. So we will form these 9 layers of retina, and the 10th layer will be the pigmented epithelium, which is also like considered as one layer of the retina. And then we are forming the lens. And how do we form lens? The posterior cells, the posterior epithelial cells will elongate. You can see how big they are. They are very big. They will form lens fibers. And lens fibers are not just like normal fibers, like collagen fibers or muscle fibers. That is a sensation. But these are just like normal elongated cells. Yes, so elongated cells. And they are forming fibers. These cells from posterior epithelium will form primary lens fibers, which will form the inner part of the lens, so the something called lens nucleus, the nucleus of the lens, but it's not a real nucleus like a nucleus of a cell, it's just like called the nucleus of the lens, which is made of primary lens fibers, which will lose nuclei. Yes, they will lose nuclei, so all you have left of these primary lens fibers are just like fibers without the nucleus. Yes, so it's called the nucleus, but these fibers don't contain nuclei. Yes, I hope you get it. And then we have anterior epithelium. Anterior lens epithelium will also proliferate, and these cells will also elongate, and they will create secondary lens fibers. The secondary ones, which you can see also in the specimen on our slides of the eye, you can also see the primary ones. And then these fibers are on the periphery, and they contain nuclei. So that's how you can differentiate these two. When you look at the lens, and you will find the fibers in the lens that contain nuclei, you can tell, okay, these are the secondary nuclei developed from the anterior epithelium. If you find the nuclei, the fibers that don't contain nuclei, you can tell, okay, these are the primary nuclei. The primary, I'm sorry, I just like completely messed that up. So once again, when you find the fibers that are without the nuclei, yes, they don't contain nuclei, they are in the center, you can tell, okay, these are the primary ones, and they are developed from posterior epithelium. Yes, the primary fibers. So I hope that's clear. Once again, you can see that on here. And what we will have left is the anterior lens epithelium. So lens actually will, in the future, we'll have only anterior lens epithelium, and this posterior epithelium will disappear. Like, it's not that it will disappear, but it will transform into these primary lens fibers. Yes, it will transform to them. That's why. So on a normal lens, in adults and children, we have only anterior lens epithelium. There are lens fibers inside, some of them contain and some don't contain nuclei, and we have also capsule around the lens, the capsule. Hyaloid vessel will slowly disintegrate because we don't need the supply of the lens actually, and that's it. Yes, and you can see that the epithelium is differentiating here. There is this neural layer and pigmented layer and mesenchyme all around. Mesenchyme all around will form the stroma of the cornea. So if you remember the structure of cornea, it's really a thick layer of dense connective tissue, very thick layer of dense connective tissue. So it's formed from the mesenchyme that is here. It's actually formed from the neurocrest. But the cornea has epithelium on its surface. Yes, the cornea has epithelium on the surface, and this stratified epithelium of the cornea, non-keratinizing stratified squamous epithelium of the cornea, is developed from the ectoderm also. So we have like 3 structures developed from the ectoderm of the eye. We have the lens, which we talked about right now, the lens. We have the surface ectoderm, the anterior ectoderm of the cornea, and also conjunctiva. Conjunctiva is also developed from the ectoderm, that is covering the sclera. Yes, and mesenchyme will give rise to the stroma of the cornea and the sclera. Yes, and what other structures are here? Yes, I mean that's, and the choroidea. Yes, the choroidea, and also the stroma of the iris and the stroma of the ciliary body. I almost forgot about ciliary body and iris. Oh God. Yes. So it's like that. This is more differentiated eye. We can see also the muscles that are forming here, the inferior rectus muscle and lateral rectus muscle. But actually, okay, we can see some of them on our specimen. That's right. The vitreous body is also forming in the inside. We have this inner layer, the outer layer, and lens. And we can also see here that the lens is filled. So fibers are filling the lens. And the mesenchyme will give rise to all structures here all around, and the stroma of ciliary body and the iris. So this is once again the thing that I talked about before. The lens nucleus is made of primary fibers, but these ones contain nuclei which will eventually disappear, and that's how we can differentiate the lens nucleus on our specimen. This is very cute, I would say. Not cute, but I like it. It's kind of strange and looks like some kind of evil monster or anything, but we can see that this is the lens with epithelium, lens, and the optic cup here, and also the optic cup with developing lens. Nice picture. So the iris, yes, yes. I don't know why do I have an iris on this situation in here, but I will tell you. Yes, these layers, the anterior epithelium, the limiting layer, and stroma iridis are actually from the mesenchyme. Yes, they are developed from the neurocrystals, and also mesoderm plays a role too in this region. And then, if you remember, we have two layers on the posterior wall. Yeah, we have posterior limiting layer, which is actually made of myoepithelial cells, which make up the dilator muscle of the eye, and we have posterior epithelial cells, the columnar cells, which are very much pigmented. Yes, so there is pigment inside of these cells, and these cells creates these two layers will create something called pars ceca retinae. So also they are like part of the retina, and as I told you before, like the whole retina is made of this optic cup. Yes, so also these layers are from the optic cup. Don't forget about it. And also we have two layers on the ciliary body. So also these two layers of the ciliary body are also pars ceca retinae, and they are also developed from the optic cup. Yes, that's very important. So not only pars optica retinae, the actual like 10-layered structure, but also pars ceca retinae is from the optic cup. Yes, and it depends like from which layer. The inner layer is developed from this pigmented layer is from the inner layer, and this dilator muscle is from the outer layer of the optic cup. So now we have just two layers, and I'm okay with that. So maybe if you have the Langman's book of embryology, there are some pictures also, but please, there are some mistakes in these pictures, because here in old pictures, in older books, this inner layer is labeled as inner unpigmented layer of iris, but as you can see, the inner layer is pigmented. Yes, so this inner layer is pigmented and should be darker. But on the ciliary body, on ciliary processes and on the corona of ciliary, the situation is correct as in this picture. So we have the pigmented epithelium, which is on the outer, that is like an outer layer. An inner layer is the non-pigmented epithelium. And now you're wondering, like, what is this labeled as an outer layer and this one as inner layer? Because it looks like that. This layer is inner and this one is outer. It looks like reversed. Really consider the layers based on the whole eye. Yes, so we have the whole eye. Oh, I'm not going to draw it, the whole eye. I'm just going to find, I hopefully will find some picture, nice picture in here. No, yes. So yes, this is the center, and everything that is closer to the center is the inner layer. So when we have two layers on here, the one that is more away from the stroma of the iris is actually the inner layer, and the one that is closer to the stroma of iris is actually the outer layer. That's the same for the ciliary body. Yes, that's the same for the retina. That's why. So I hope you're not confused at this time right now. Sorry for these images. Here, I'm back to my previous position. So yes, so please note that the inner pigmented layer is from the inner layer of the optic cup. It's pigmented in iris, but it's not pigmented on the ciliary body, because these cells on the ciliary body, they do not contain pigmented granules, or maybe a very small amount, and they are producing aqueous humor, they are producing fluid, because ciliary body is responsible for accommodation of the lens, and also the ciliary body is responsible for the fluid production in the eye. So these cells are developed also from the inner layer of optic cup. And the stroma of the ciliary body and iris is from the mesenchyme, except for the muscles. So you know the dilator muscle is from the outer myoepithelial layer of iris, could be pigmented also, but also sphincter muscle is from the neuroectoderm. So all muscles of the iris are from the neuroectoderm, from the optic cup. And this is the picture of the retina, the pars optica of the retina. We can see some layers, and I'm not going to describe them, like you should know them, but don't forget the light. The sun is going all through these layers to the pigmented epithelium that is here. Then we get to the rods and cones, bipolar neurons in this layer, bipolar neurons and ganglionic neurons. Yes, we have less ganglionic neurons because many bipolar neurons are connected to maybe one ganglionic neuron. And then we have nerve fibers going outside to the optic cup. So, pigmented epithelium, this epithelium here is from the outer layer, and these layers are from inner layer of optic cup. Okay, so yes, it's like that. So that's why on our specimen, on our slide, you can see sometimes that these cones and rods and all these layers are separated from the pigmented epithelium, and it's pretty much normal because there are no connections in between. Yes, there are no connections in between, and so they are separated. So how can we hold the retina, these two layers? How can we keep the intraretinal space closed? Is that we have the vitreous body in here that pushes the retina, these 9 layers, to the pigmented epithelium, so they are close together. Yes, and we have, this is the iris, this structure in here is the iris, and this is the ciliary body. And you can see the the cells are reversed here because pigmented layer in here is the inner one because it's closer to the center of the eye. So the inner one is pigmented, and on the ciliary body this pigmented layer is the outer one. So we cannot say it like that the pigmented layer is developed from the outer layer, because it's true for the ciliary body, it's true for the retina, the pars optica, but it's really not true for the iris because this pigmented layer is from the inner part. So it's much more complicated, as always, like, you know. And we have, what can we see here in the lens? We can see the nuclei inside these fibers, lens fibers. So these are the secondary fibers. Inside there are primary fibers, no nuclei in here. We can see anterior epithelium, like simple cuboidal epithelium, and we can also see this capsule, which is all around the lens. The capsule is all around, but this epithelium is only on the anterior part. Yes, so once again, you can see that the conjunctiva sac is forming here. We haven't been talking about that, but it's forming by invagination of the surface ectoderm. So the conjunctiva is from the ectoderm, and the anterior epithelium of cornea is from the ectoderm. The stroma is from the mesenchyme, yes, and the sclera is from the mesenchyme also. Don't forget that optic nerve is covered with the dura, because right now we are in the central nervous system. So the sclera is continuous with the dura, which is one of the meninges, and also there is a pia next to the optic nerve. So another picture, the epithelium is more developed here, everything is more developed. It's a really nice picture. And I don't know like what to do and how to describe this picture right now, but I hope it's pretty straightforward. Yes, so lens, ectoderm, the cup, the neuroectoderm, we talked about it, the retina, and everything else, other structures that are there are from mesenchyme and surrounding mesoderm that is there. First, there is a membrane that is developed and it's covering the whole eye, the membrane. The papillary membrane will cover the whole eye, but eventually it will rupture and it will form the pupil between the iris. Yes, inside the iris, I mean, between these two structures, so inside the iris, this membrane will rupture. So just remember, first the mesenchyme will go through the whole eye on the anterior surface. It looks like the eye is closed completely, but then it will rupture on the inside, forming the pupil. And this is how it looks like in this. We have the embryos developments. We can see the black coats. In here we can actually see the forming eye inside, and this is normally the fetus. Let's proceed to development of the ear. Ear is composed of three basic parts. We have external ear, we have inner ear, and the middle ear in between. And you know something about external and middle ear, I suppose, because we have covered pharyngeal arches. And you know there are some structures like pharyngeal clefts and pharyngeal pouches, which are in between of the pharyngeal arches. So I'm going to show you this picture maybe. So you know, like many structures are developed from pharyngeal arches, and we have like 4 pharyngeal pouches which will turn into parathyroid glands, the thymus, the palatine tonsil, and you know, like stuff like that. But let's cover the first pharyngeal pouch. The first pharyngeal pouch will form a tubotympanic recess. Tubotympanic recess, which will transform into, like this name is telling you what will it transform into. It will transform into tympanic cavity and the eustachian tube, the auditory tube, actually. Yes, this tubotympanic recess. So from the first pharyngeal pouch, which is covered with endoderm, we will form the inner ear, the tympanic cavity, and eustachian tube. From the first pharyngeal cleft, we will form external ear. And what is in between the external ear and inner ear? Where the eardrum is in between, which will form from the mesenchyme that is here from the neural crest. Yes, so mesenchyme that is here all around. There is also, once again, like normal mesoderm also here, but we have eardrum from the mesenchyme all around forming in between. Yes, so there is no hole in between these two structures. There is this eardrum. Okay, so we have external ear and middle ear. But what about inner ear? How do we form inner ear? Actually, first we need to form an otic black coat. Yes, we have also like lens black coat. So now we have otic black coat, which is actually a thickening of ectoderm. It is at the level of neural tube, so we have neural tube over here, and at the level of future developing hindbrain, we will form these two thickenings on each side of the head, the otic black coats, thickenings of ectoderm, and we will form the inner ear. And as you probably know, the inner ear is inside of the head. Yes, so this otic black coat, although it's formed on the surface, it needs to go inside. And how are we going to do that? This otic black coat will invaginate and we will create the otic pit. Yes, and these cells will proliferate on this otic black coat and they will invaginate, creating the otic pit. And this connection with the outside space will close eventually and we will form something called otic vesicle. Otic vesicle. So it's just like the lens vesicle, yes. It's like also the same. We created lens like this way, but now we are creating inner ear. Yes, and some structures are going to form from this otic vesicle, but there is another structure called stereoacoustic ganglion or vestibulocochlear ganglion, which is also going to form from this ectoderm. Because these cells of otic vesicle will delaminate, they will just separate. Yes, let's call it a separation from this epithelium, and they will form the ganglion. Now you're just probably thinking like, okay, but we learned that the ganglia are from the neural crest. Like in general, yes, all ganglia are from the neural crest, but vestibulocochlear ganglion is from the ectoderm. Yes, so it's like that. There are some nice pictures. Yes, I like especially this one when we have the otic vesicles in here. Yes, the otic vesicles. We have the tubotympanic recesses in here, and yes, and future aortas maybe in here. At least I hope so. And here is forming neural tube. It's not closed here. Oh, maybe it's not. It is just like the thin membrane in here. So it's probably at the level of the mesencephalon. Yes, the fourth ventricle here. And it actually makes sense because very close to the hindbrain we will have the otic black coats. Yes, and this is what I described earlier. Lens black coat, otic black coat. Yes, two black coats which will form the structures that I have described earlier. Yep, this is once again the otocyst in here, the ganglion. The ganglion once again is derived from the ectoderm, which is something strange because normal ganglia are from the neural crest, but this one isn't. But the nerves and glial cells, actually the ganglion itself is from the ectoderm, but nerves of the vestibulocochlear nerve and glial cells of the vestibulocochlear nerve are from the neural crest. So there you have it. The neural crest says it best. So the nerves and glial cells are from the neural crest. Yes, and this otic vesicle is somehow going to differentiate into some structures. So what do we have here? It is going to form the endolymphatic duct, or first the endolymphatic appendage. So from this vesicle we have some outpouching, so it's going to divide. This part will expand and it will form the pars superior, I would call it first, the pars superior, and this part in here, the pars inferior, like of course we have pars superior and inferior, and in the lymphatic appendage, then in the sac, and then we call it the duct. And from these structures, which are from the ectoderm, we will form some structures of the inner ear. It's about in the 4th to 7th weeks of development. The pars superior will, once again something is happening. The pars superior will transform or will differentiate into 3 semicircular vesicles, channels or canals. Yes, this pars superior will give rise to semicircular canals, and the auricular region, the inferior part elongates and really give rise to the saccule, but it's not written here, but also the cochlear duct, which is inside the cochlear duct. But all of these structures that I described, they have some bony part and some membranous part. We have like bony labyrinth and membranous labyrinth. So only membranous labyrinth inside the inner ear is developed from this ectoderm. Please remember that, membranous labyrinth. Yes, because bones are eventually not from ectoderm, they are from the mesenchyme that is all around. Yes, so only membranous part, that is like epithelium that is there, is from the ectoderm, from this otic vesicle. The tubotympanic recess will go close here and then needs to somehow communicate with this sac that is developing here and dividing into some parts. And you can see something is happening here. These pink structures are ossicles, and they will condensate and they will form malleus, incus, and stapes. Malleus, incus, stapes, the 3 bones that are forming inside the tympanic cavity. But right now they are not inside any cavity, they are just normally in the mesenchyme, like forming, like normally bones are formed in the mesenchyme. Yes, so now on we don't actually have tympanic cavity. So what is going to happen? As you know, the 3 auditory ossicles, they will develop from this mesenchyme adjacent to tympanic cavity, but they are from they are actually from the mesenchyme of the 1st and the 2nd pharyngeal arch. Yes, so the malleus and incus, I don't know if I have picture somewhere. Yes, so the malleus and incus are from the 1st pharyngeal arch, mesenchyme. As you remember the Meckel's cartilage in the first pharyngeal arch, the pharyngeal arch mesenchyme, and the stapes is a second arch derivative. It's derived from the second arch. Yes, so malleus incus are from the first, and the stapes is from the second pharyngeal arch. But first they are like condensated in mesenchyme, and the second, the tubotympanic cavity, will enclose these bones inside the cavity. So these bones, yes, this recess is going closer and closer and will go all around these bones, so they will actually be covered with the endoderm. Yes, so the cavity will be all around and they will be also covered with the endoderm. Like it's like that the cavity will just like engulf these ossicles in here. Yes, and that's how we can connect the inner ear, the like external ear, the inner ear, the middle ear together. Yes. Once again, the malleus incus. First, they are covered with the endoderm of the recess, because if they were like engulfed with the tubotympanic recess, and in this recess, in the epithelium of the recess, we will form ligaments, because we need ligaments to attach these ossicles. Yes, and the cavity will be inside, and now they are not covered with the endoderm at all. Only they are inside the cavity. Once again, Meckel's cartilage is very important in development of malleus and incus. Yes, so they are like the origin of these two is from enchondral ossification, like the chondrogenic ossification, and the stapes is from the second pharyngeal arch. Yes, from there is Reichter's cartilage in the second pharyngeal arch. So, please don't forget about these ossicles, these three ossicles. You need to know how they are developed. Also, don't forget about the pharyngeal cleft. Yes, the first pharyngeal cleft will give rise to external ear. The middle ear is from the pharyngeal pouch, from tubotympanic recess. The ossicles are from the mesenchyme that is condensating all in here in the first and second pharyngeal arch. And then we have from surface ectoderm, we develop the inner ear, and everything that gives rise to membranous labyrinth inside the inner ear, and the bony labyrinth, the bony structures all around, and the tissue all around is of a mesenchymal origin. The ganglion, the vestibulocochlear ganglion, is from the ectoderm, but the nerves and glial cells are from the neural crest. And we need to form also the auricle, the pinna, and the external ear, the pinna and the auricle is developed from the 6 auricular hillocks. So 6 hillocks will form the auricle, and this actually makes sense for the animals more, but for the human it seems like it's very difficult to develop the auricles, because it's like very strange structure, yeah, but it makes much more sense in animals. So you don't have to know specifically things that are developed from each auricle, like don't learn it that way. Just know that there are some auricles that are from the lateral edges of the first and second pharyngeal arches. Yes, so lateral edges of the first and second pharyngeal arches will form these auricles here. And this is how the ear looks like, in case you don't know how it looks like. Yes. And the last thing we are going to discuss is olfactory. It's not like the last thing at the end, but we're going to discuss the olfactory epithelium development. You just need to revise if you don't remember the frontal nasal prominence, which will create these nasal plackets here. So we have also nasal plackets. So the frontal nasal prominence will create the nasal plackets which have medial and lateral prominences, and inside there is a nasal pit. The olfactory epithelium is actually developed as an outparching also from the prosencephalon. Yes, so olfactory bulb is also from the prosencephalon connected to the forming nasal cavity. Okay, the sensory cells that are here are primary neurosensory cells, primary ones. We will talk about it. The primary neurosensory cells means that these cells are actually neurons that are transformed into these special cells. That is also the case of the rods and cones in the retina, but it's also the case with these olfactory special cells. And now let's move to some other sense organs. Like this was the development, but now really let's talk about the structure of some sense organs. So how can we divide the receptors of sense organs? Please be aware of this morphological classification. It's very important for us, and we're always like ask you during examination, okay, like this is the sensory cell, but is it a primary one or secondary one? What do you think? Like all the time. That's the questions that are like very common. So when you are talking about some sensory cells, you should be able to tell like if it's a primary receptor, that means that these cells are modified neurons and actually are developed from some neural epithelial layer. Or if these cells are secondary receptors, that means they're like normal sensory cells and they are modified epithelial cells. Modified in that way, they are able to somehow transform the signal to the neural cells, and yes, it's like that. They are not real neurons. They don't have axons and dendrites. Yes, they are just like epithelial cells which can transform the signal to the neural cells. And then we have these peripheral afferent nerve terminals, which can be encapsulated or free. Yes, we have many types of encapsulated afferent nerve terminals, and they are for the proprioception, the touch, and heat and cold, and for the pain. Yes, so we're going to talk about them. Let's start with olfactory epithelium. But not in this case. Yes, I can see some rods and cones. It's just like I wanted to explain in this number 1 slide that these are the primary neurons. Yes, and we will start with olfactory epithelium eventually, but the cones and rods and the olfactory cells are primary neurons. They really are neural cells. That means that they have some processes of neurons as well. They have dendrites and they have axons. Yes. So, for example, these olfactory cells, they have dendrites in here. They are going like outside of the epithelium on the periphery, and they have also axons, which are going to the deeper layers. Yes, and to the brain. Okay. And then cones and rods, for example, so these are very modified dendrites actually. Outer segments, the photosensitive region. I don't know if we can call them dendrites anymore. I think not. Just like call them the outer segments of cones and rods, but they do have axons on the other surface. Yes, so axons are the other surface, and in external plexiform layer there are many synapses with bipolar cells connecting together. So bipolar cells are releasing dendrites, and these dendrites are connecting with the axons of the cones and the rods. Second, these are the secondary sensory cells. So we can see the taste bud. Yes, taste buds in here, we're going to describe that, or some hairy cells in the ear, hair cells in the ear, So also these are the secondary sensory cells. And then the third group are the afferent nerve endings, the terminal nerve endings, and we are creating these Pacinian corpuscles or Meissner's bodies, Ruffini free endings, Krause ones, and also a muscle spindle and Golgi tendon organs are very important. They are going to the spinal ganglion because they transform the sensory signals to the spinal ganglion, and this is actually really the spinal ganglion. I hope you know that, because we can see the very big neurons in here with continuous sheath of satellite cells all around, so we know that these cells are pseudo unipolar cells, and we can see different sizes. So these are the very big ones, and these are very small ones containing the nucleus. Yes, you can see this is very small neuron and this one is very big. So these are typical features for the spinal ganglion actually. So let's start with olfactory epithelium. You should be able to draw these cells. Yes, so during examination they will probably ask you, like, can you draw these cells? They are like very special, they have very special structures. So these cells, don't forget that these are the primary sensory neurons, and they have, I will draw it like this, they have the thick body inside because they contain a nucleus inside, well, of course, and then dendrite is going to the periphery of this epithelium on the outer surface here, and this dendrite, it will create this like white structure like the olfactory tip here, which contains cilia. These cilia are really perceiving signals. They are not moving like normal cilia in respiratory epithelium. They are not moving, but they are there. So this is very special, like the dendrite has this widening on the apical portion and also contains cilia. Yes, and they are very much modified. And they also have an axon. So they have an axon, which is going to deeper layers of the nasal cavity to the lamina propria. You can find this type of epithelium in the nasal cavity on one of our slides. This olfactory epithelium, it's not just composed of these cells, these olfactory cells, but there are also sustentacular cells. They are like helper cells. They look like this, helper cells with microvilli on their surface. Yes, so they have microvilli. And so the outer nuclei of this epithelium belongs to sustentacular cells, and the deeper nuclei belongs to olfactory cells, the sensory cells, and also we have basal cells. So there are also basal cells which will differentiate, they can divide, and they can form these neuronal cells in here. And this is how the epithelium looks like. So this is the septum in the middle. We have the respiratory epithelium here, but like don't forget that the respiration is not the case in here, so no gas exchange in this place, only in the lungs. And the epithelium is now higher and higher, and it's very high, and it's creating this olfactory epithelium. So olfactory epithelium is a pseudo-stratified epithelium with cilia, also as the respiratory epithelium. But what are the differences? It's much higher. Well, we can see that it's much higher. It's also in this place over here. And this is the concha, the nasal concha. And so it's much higher. It contains cilia, but these cilia are not moving. Yes, they are not moving. And there are also sustentacular cells. They are these olfactory cells with cilia. Yes, so the composition of this cell is of these cells in here is very different than the composition of the cells of respiratory epithelium. And also, what are the other differences? So we can see the epithelium, yes, but also there is a lamina propria in this place under epithelium, and under olfactory epithelium, the lamina propria contains special glands that are called Bowman's glands. Bowman's glands. I know this picture isn't very nice, I am sorry, but you can go to the slide room and try to look at the nasal cavity, and you will see the Bowman's glands that are in there. Yes, or I can take you another picture, but this isn't a very good picture. Maybe here you can see it better. These glands are Bowman's glands. They are serous glands. Yes, serous branched glands that are in here, they have ducts. They go right through the whole epithelium, and these ducts open at the surface of the epithelium. They will release the fluid which will wash things away from these epithelium. Yeah, so you can smell different things at the time. Yes, so you can smell something nice in a minute, and then you will smell something that smells very bad. Yes, and it's because of the Bowman's glands that are washing the surface area for the new sensory stimuli that is there. Also, what we can find in the lamina propria under the epithelium are the nerve fibers. Yes, we can find many nerve fibers in these lamina propria, because these nerve fibers are actually axons which are going from this epithelium, from the olfactory cells. Yes, this is the epithelium. So I would say that these nuclei that are here on the top, on this portion, these nuclei belong to sustentacular cells. These nuclei in here belong to olfactory cells, and these nuclei, which are really in the base, belongs to basal cells. So don't forget about 3 types of cells that are in here. And these are the cilia on the surface, and the microvilli on the surface. And the Bowman's gland in here. And actually I cannot see the fibers, maybe these ones, but we cannot see that in the picture. Don't forget, these are like the part of peripheral nervous system, these like axons that are going. So they are covered from Schwann cells in this lamina propria. They are from the Schwann cells. Yes, it's also written here, Schwann cells. Okay. So supporting cells or sustentacular cells, that's the same. Like you can call them supporting cells. That's pretty much okay. I'm okay with that. And you should be okay too. So, okay. So the cilia have very similar structure. The axonema has very similar structure to normal cilia, but it's thicker on this proximal part, and on the distal part is much more thinner. As you can see, only some of microtubules make up this part of the cilium, but they are not moving. That's all. They're just like receptors. Yes, they're on the membrane, but they are not moving. This is picture from the oral examination. So you should differentiate the olfactory knob in here, olfactory knob with cilia on the surface. Yes, cilia. So many cilia on one knob. It's like widening of the dendrite actually. And they are also microvilli all around. So these structures are microvilli. And these microvilli are from the supporting cells, the sustentacular cells. So this is one picture from the oral examination, and you should know that the cilia are not moving. What are the specifications of the olfactory epithelium? Which cells are also there? Yes, so don't forget about this stuff. It's very important. And the Bowman's glands, are serous branch glands which have their ducts going right through the epithelium on the surface. And you can see from olfactory cells, there are axons going, and these axons together, if you find them under epithelium, like on our slides, you can see like many nerve fibers going from this epithelium on in lamina propria. They are called phyla olfactoria. Yes, these are the Bowman's glands in here, and these are the cells of epithelium. And I don't know if these, why is this labeled with C and D, I'm not sure, but yes, these are the phyla olfactoria. Yes, the phyla olfactoria, the nerve fibers in here. And wow, in this picture we can see nicely the Bowman glands and the duct. So what about the taste? Taste, we have taste buds for the taste. Yay! And taste buds are made of many cells. We differentiate dark supporting cells and light sensory cells. So the sensory cells that are actually lighter are the sensory cells, yes, and the dark are the supporting ones, and also there are some basal cells. So from the basal cells, we can create these dark supporting ones and light sensory cells. Taste buds and these sensory cells are secondary cells. That means they are not transformed neurons. They are just like normal transformed epithelial cells connected to the nerve fibers. So as you can see in here, there are peripheral branches of axon of an afferent neuron that are going here. So they don't have axons actually, these cells. They are like normal epithelial cells. This is the taste pore, which you can actually see on some slides, on some specimens. You can see on some slides also the taste buds. And you probably know these taste buds are located all around the oral cavity on the tongue, mainly on the velate papillae. Yeah, many of them are on velate papillae, but they can actually be found on many other papillae in the tongue except from filiform papillae. So we cannot find them on filiform papillae, but on the other papillae it's quite good. But most of them are on velate papillae because these are the biggest papillae, so they contain many taste buds. You don't have to differentiate these cells in the taste bud. Just know that like there are many types of cells, like maybe know about these three basic cell types, but you don't have to differentiate them because it's very hard to find the taste buds, not just like differentiate the cells that are in there. So be okay with that. They contain different receptors, different channels. They are from different taste. But remember, all taste buds can taste all tastes. Tastes. Yes. So one taste bud is able to differentiate the bitter, the sweet, the umami, and the salt and the sour at the same time. Yes. So it's not like that some taste buds are for they are for sweet and some of them are for the salt. It's not like that. This is the taste bud in the epithelium. It really looks like just some kind of papilla from the lamina propria, but it's not. But you will be able to differentiate them. Don't worry. Just find a slide with the tongue, yes, and try to find some taste buds. So we can see nicely in this surface stratified squamous epithelium, this round white structure. There is the pore. Oh, sorry. My pen is broken. Okay, once again here the pore, and this is the taste bud. In here you can see some cells are inside. Maybe these cells in the basal portions are the basal cells, and we have light and dark cells inside. Yes, so the food is somewhere in here, and with the microvilli on the surface we have these receptors on there and we can taste. And here, somewhere here, but we cannot see that, are nerve endings that are connected with the cells inside the taste bud. And once again here, the pore. The pore is washed with the secretion of glands that can be found in the on the tongue. So Ebner's glands, I hope you remember, there are von Ebner's glands and Weber's glands. Yes, and these glands are washing these taste buds, and so you can taste different tastes. I hope you get what I'm trying to say. It's as the same as for the olfactory epithelium. You need to wash the surface epithelium all the time. Yes, and this is a picture from electron microscope. Many types of cells, not so important. So let's proceed to some other structures, like for example, the glomus caroticum is like from the taste to glomus caroticum, yes, or the carotid body. It's called caroticum because this structure is located very close to carotid arteries, and we don't have a slide actually for this. So I wouldn't say it's not that important, but like you should know something about it, but we actually don't have a slide from this structure. So it's located near carotid arteries, yes, in branching of the common carotid artery into internal and external one. Yes, this one, external, internal, in the carotid sinus, near the carotid sinus. And the glomus, it's actually accumulation of ganglionic nerve cells that are very sensitive to oxygen levels and pH levels. Yes, especially also carbon dioxide levels, but very sensitive to oxygen levels. This is how it looks like actually. So we have some kind of artery in here, some probably carotid artery or some branch of carotid artery, and we have accumulation of neurons very close to that, and these are the ganglionic cell of glomus caroticum. We have also the sinus caroticus. The sinus it really looks like a widening of the artery. I'm sorry, I'm struggling with speaking. So the sinus caroticus, it's really just you would see what we will see on histology specimen. We will see the narrowing of tunica media. So the tunica media is narrowed pretty much, and widening of tunica adventitia. Yes, so tunica adventitia will widen, and this tunica adventitia will contain many nerve endings. And sinus caroticus is a baroreceptor. So, oops, I'm sorry. So baroreceptor, it is true. And this baroreceptor is for the how to say it, like for the pressure. So when there is a high pressure, the carotid artery will widen. Yes, it will like stretch a lot. So these nerve endings which are in the adventitia will react to that. Yes, that's why we have a thin layer of tunica media. So when there is like a higher pressure in the carotid arteries, this structure will widen even more than the whole artery because the tunica media is thinner in this region. And now let's proceed to proprioceptors. So the muscle spindle is actually on our specimen, so we'll describe the muscle spindle even more. The Golgi tendon organ, it's pretty much fine, but Golgi tendon organ is it's much smaller than the muscle spindle. It's encapsulated structure, and really what these are the sensory axons, and these sensory axons are penetrating through the collagen bundles of the tendon. Yes, so they are Golgi tendon organs. That means they are in the tendon. These axons are penetrating to the tendon, and they are inhibiting motor nerve activity. They are inhibiting because when there is a muscle contraction, and it's like very, very big muscle contractions, you don't want your muscles to rupture or something. So these Golgi tendon organs will inhibit motor neurons, so they will lower the muscular force. Yes, so that's why we need them. Something different are these muscle spindles. Muscle spindles are very complex structures. So once again, we have afferent nerve endings that are all around the muscle fibers. So muscle fibers are the nerve endings are all around the muscle fibers. We call them intrafusal fibers. I will show you the pictures. And they will wrap, these axons will wrap around individual fibers. And what is going to happen is that there are also extrafusal fibers all around. So these organs, the muscle spindles, are really actually in the muscle. And when there are changes in the length, like distension of the muscles that are all around, these distensions, the changes in length, are detected by the muscle spindles, and these sensory sensory nerves that are all around here will relay the information. They will relay the signal to the spinal cord when there is stretching, too much stretching. Yes, so when there is a stretching, your spinal cord will be like, oh, there's a stretching, we need to do the contraction. Yes, so the spinal cord will then send the signals to the extrafusal muscles to contract. So it's very active proprioception. This is how the muscle spindles looks like. So we have them also on our specimen. They contain intrafusal muscle fibers inside. That just means like muscle fibers inside the muscle spindle. Yeah, it's not that complicated. Just muscle fibers inside are called intrafusal. Muscle fibers that are outside are called extrafusal. It doesn't mean that extrafusal muscles are part of the muscle spindle. It's not like that. Extrafusal fibers are just fibers that are normally in the muscle, in the skeletal muscle. But somewhere in the skeletal muscle, like in all muscles, most of them, there are these muscle spindles, yes? And these muscle spindles are encapsulated organs, so there is some fibrous tissue all around. And we have intrafusal fibers in here, intrafusal fibers, and the nerve endings are wrapped around these intrafusal fibers and are detecting the changes of the length of these intrafusal fibers. Because when this muscle, when these extrafusal muscles will stretch, these intrafusal muscles will also stretch. And these nerve endings around these smaller interfusal fibers will send signal to the spinal cord that the muscles are stretching. Yes, it's like that, because we don't want the receptors on each muscle fibers in here, so we developed this small structure that is also changing related to the extrafusal fibers. There is also a fluid inside in here, some fluid. It's like normal interstitial fluid, and there is a capsule, the fibrous capsule around. This is how it looks like on our specimen. So sometimes you will get the slides with just like with the muscle tissue, the skeletal muscle tissues, and you will be just fine. Okay, I got the skeletal muscle tissue. Pay attention and try to find this little structure in here. We will do that on our practical trainings in the last week. So try to find the muscle spindle. Once again, these are the intrafusal fibers, just the muscle fibers, normal muscle fibers, but they are smaller. We have also this inner capsule, and then we have outer capsule all around. And all around the intrafusal fibers, we have nerve endings wrapped around, which are sending signals to the spinal cord. And these muscles all around here are extrafusal fibers. Let's skip to other receptors. And what do we have here and here? So, these are Vater Pacinian's corpuscles. Uh, like large oval structures like this. These are the Pacinian corpuscles. They are much more smaller on our specimen. It's very sad that we have like very small Pacinian corpuscles on our specimen. They should be approximately like 1 millimeter big. They are so big. They are found in a hypodermis and dermis also. They can be found in a hypodermis and dermis. And they have outer capsule. There is an outer fibrous capsule in here. In the middle, we can find nerve endings, afferent nerve endings. And you can see the several layers that are in here. And these layers are composed of flattened Schwann cells and perineural cells. Yes, so like modified fibroblasts, but also there are Schwann cells. First, we have some layers of Schwann cells, and then we have some perineural cells. We cannot differentiate them. We just see the lamellae that are in here. So many lamellae are creating these Vater Pacinian bodies. They are like 250 lamellae actually in these bodies. Yes, so they are important for the sensation of the pressure. Yes, so they can be found in connective tissue of organs, also organs located in deeper parts of the body, but also in the fingers of your hands. Yes, so they are mostly like everywhere in the body, and they are important for the sensation of the pressure. Pacinian, Vater Pacinian bodies or corpuscles. Then we have Meissner's corpuscles. Meissner's corpuscles are ellipsoidal structures or elliptical structures, and they are composed of axons, but we cannot actually see the axons. Maybe we know they are branching somewhere inside. This is like the terminal ending of the axons here. And they are also Schwann cells there. And these cells are Schwann cells. We can see the nuclei, and they are organized in a perpendicular pathway, or how to say they are organized like in perpendicular fashion. Yes, like this. Okay, perpendicular to this axis. And where are we right now? Could you recognize the tissue? Where are we? Yes, we are in the skin. The thick skin, actually, because we can see the stratum corneum is very thick and all layers are very thick. So the epidermal thickness is what can tell us that this is like the thick skin. And in the stratum papillare, the papillary stratum, in the loose connective tissue actually, below the epidermis, we can find Meissner's corpuscles. So once again, just axons are inside with Schwann cells organized in this fashion. Yeah, the Schwann cells are perpendicular, so it's really like hard to recognize them sometimes, but they look like this. There are many cells organized in this fashion in one place, and that is suspicious for the Meissner's corpuscles. They really are responsible for light touch, yes, or like very low frequency stimuli, but so they can deform their shape. We can find them in the fingertips and palms, yes, because we can see there is a thick skin in here. So they are in the palms and soles, and many of them are in the fingertips, and that's actually it. Like, you don't have to know really the differences between like these ones are for the low frequency like stimuli, and these are for the high frequency or the fine touch and stuff like this. Like, all these corpuscles are important for the touch sensation in some kind of way, the pressure sensation, but you don't really have to know the differences. Actually, you don't need to know like how do they look like, how can we find them in the tissues, yes. And they are, all of them are for the touch sensation, also for the heat and the pressure, because all of them can do all the stuff, but some of them are more specialized for some stuff. Yes, so for example, we can see a Ruffini corpuscle, but we don't have specimen, and you don't have to differentiate Ruffini corpuscle, but actually once again it's encapsulated axons that are here, and you probably know Ruffini corpuscle that is important for the sensation of the heat. Yes, heat, but also Ruffini corpuscles we can feel pressure because of them. Yes, so it's not just about heat. So you should know the structure of Pacinian corpuscle, the Meissner's corpuscle, and also there exists this thing, the Merkel's disc. Merkel's disc, you probably know the Merkel cells. Merkel cells are cells in the basal layer of the epidermis, very light cells that look like melanocytes. They look like melanocytes, but and they are afferent sensory endings, very close to Merkel's cells, and that's why this is called Merkel's disc. Yes, so they also are important for the pressure sensation. Yes, so for example, Ruffini endings are for the skin stretching and also heat more. The light touch sensation is going through the Merkel's disc. The vibration is in Pacinian corpuscles and also Krause. I don't know if it's written in here, but also Krause corpuscles exist. Yes, and also these Krause corpuscles are very important for the low frequency vibrations in the penis and the clitoris. Yes, so they are in the penis and clitoris, so that's why they're important, and they are for the vibrations. So also Pacinian corpuscles are for the vibrations, but they are also for the touch, yes, and pressure, but they are more sensitive to vibrations, and heavy pressure is transmitted via the Meissner's corpuscle, for example. Yes, but they can feel everything, most of it, with all the discs and the corpuscles in here. Okay, and that's the thing. That's the end of my presentation. I hope you will do well during examination. I wish you good luck and good summer and everything. So I hope this helps you, and goodbye.